Okay, this is where we finished last time. This is the third and final prep for lecture 15. And I usually try to keep the preps shorter, but I think going over this one a little more slowly and carefully is probably going to be helpful. So I want to just reiterate, I say it over and over because it's a common, not because I think, there's anything deficient in your understanding. It's just that traditionally over the years, this is the kind of terminology that most students have found somewhat confusing. And so I would like to try and avoid that if possible. So just a reminder that, we have the parental generation here. It shows two linked genes. This parent, both homozygous for the dominants, this one homozygous for the recessives. The F1 are going to have one chromosome from each of those parents. And I've done the color coding deliberately to try and show that. And when we look at their gametes, the gametes they produce, the parental ones have come from one parent, have gone through this individual, and then are passed on unchanged with respect to those two genes, at least. They're passed on unchanged from the grandparent. Essentially, this will be passed down to the grandchild. Same with this. This section of this chromosome is passed to this individual, and then if there's no crossing over, it's passed unchanged to the next generation. I have a friend who did the 23andMe, where you look at your DNA, and she had DNA from her mother as well, and so she could look at the places along a chromosome that came from her father and came from her mother. It was it was a very interesting thing actually for her to see, and that chromosome would be non-parental. Okay, and so if you look at the DNA level, you can see this, but if you look just at schematic level, you can see it as well. These are non-parental gametes because they have portions of the chromosome that came from one and the other of the parents. Okay, so all this ink stuff is a mess, but hopefully that made it sort of clear. All right, and so we again, the calculation of map distance is actually a real thing, and sometimes students will kind of put the map together in a way that sort of isn't logical because they, for example, they'll put the genes alphabetically, A, then B, then C, even if the map distances mean that the order shouldn't be like that. And so I just want to reiterate that it isn't just an exercise. It's a real thing that's being calculated, and the map units tell us approximately where on a chromosome the genes are located. Okay, and so suppose you've done, we can use those map distances to figure out also the arrangement of genes, because if you calculated the map distance between gene A and B, and it turned out to be 5 map units, and the genes A and C was 1 map unit, you would have 2 different possible arrangements. A could be between B and C, or C could be between B and A. And how would you figure that out? You would figure out the distance between genes B and C and find out if it was 4 map units or 6 map units, and that would allow you to determine the order. When we say linkage arrangement, we're talking about the order of the genes along the chromosome and then the distances between them. Okay, so I'm going to go through hopefully some examples. We're going to look at vestigial and purple, which we've talked about before, we already know are on the same chromosome, but we're going to go through all the crosses. So the genes that we're looking at are an allele for the black body color instead of the tan color that flies normally have. The wild type is B with the the plus, the wild type superscript. Vestigial stubbly wings that are shorter than normal compared to the normal wings, and purple eyes recessive to the wild type, the normal dull red color. And so we're going to do 3 sets of crosses each pair of genes so that we can look at black, purple, and what's the other one, vestigial, and find out what their order is and what the relationship is to the other 2. Okay, so 3 sets of crosses. So the first one is a parent that's homozygous mutant for black and vestigial, and then one that's wild type for black and vestigial. So that's a cis combination with the 2 dominants together on the homolog. The wild type heterozygotes have this arrangement. And then when we look at the progeny from this cross, we see 822 that have both wild types, 652 that have both of the mutants, and then 130 that are wild type for black, mutant for vestigial, and 161 mutant for black and wild type for vestigial. So when we look at these numbers, there's no question these are our 2 non-parental combinations, and notice that these are reciprocal classes. So this one has the wild type allele for black, but it doesn't have the mutant allele. This category does. This category has the mutant allele for vestigial, but doesn't have the wild type, but this category does. So when we combine the 2 reciprocal categories, they contain both alleles of each gene, although not necessarily in the same orientation, arrangement that they were in the parents. And that's what reciprocal class means. So if you add the 2 alleles for this parental class and this parental class, there's 1 mutant and 1 recessive allele for both of them. And we can figure out the R value by taking 291, which is the recombinant ones, divide by the total, 1765, and that will give us an R value of 0.165 or 16.5 map units. When you're calculating map units, you should round to just 1 decimal place. Sometimes the calculation will give you like 6 or 7 different decimal points, but think about something like your height. I am 70 inches tall. I could, if I was 70.5 inches, that might be meaningful, but 70.53169 inches is kind of a meaningless thing to add those extra decimal values. So we're going to keep it sensible and just keep it at 1 decimal place. Okay, so that's the first one, 16.5 approximately map units between those 2 genes. Now we do black and purple. This time we have a trans heterozygote that's produced. Black parent, mutant for black, but wild type for purple. And then the purple parent, purple eyes, but wild type body. And then when we look at the progeny classes, the parental ones being mutant for black, but wild type for purple, or wild type for black but mutant for purple are the larger categories. Those are our parentals. And there are not very many recombinants, only 52 that are wild type for both and only 40 that are mutant for both. And the reason why the numbers are kind of wonky, not wonky, but they're not out of like exactly 1,000, is because these are the results of real crosses, and these are the kinds of numbers that were generated. In an exam question, especially midterms, I am aware of time limitations, and if I make things out of 1,000, for example, you can often do the arithmetic part of it in your head, and it'll be easier and quicker. So you will probably not see numbers like this on exams, but you might on practice questions and practice assignments, because we give you a lot more time to work on those. Okay, so let's calculate R. R is going to be the recombinant ones, which is 40 plus 52, so 92 over the total, and that gives us 6 map units. Okay. So now we know 2 of the numbers. So we know that black and vestigial 16.5 map units apart, black and purple 6 map units apart. So there's 2 possible combinations. Black and vestigial could be on the outside with purple in the middle, and the arrangement would look like that with 16.5 for this. And in that case, we could figure out, we would expect the vestigial to purple distance to be around 10.5 map units, right? But it could also be this combination with purple and black 6 map units apart, and then black in the middle, and vestigial black distance is 16.5, and so this would be a total of 22.5 map units, right? So when we map purple relative to vestigial, the number that we generate is going to tell us, oh no, not this one, but this one is going to tell us what the actual arrangement of them is. We know vestigial can't be in the middle because purple is closer to black than vestigial is, and so there's no way to make these two numbers work with vestigial in the middle, okay? Because black and purple would have to be on the outside, and this number would be smaller than this distance, right? It wouldn't work. So let's just show that black. Or sorry, vestigial, what am I saying? Vestigial in the middle, vestigial to black, vestigial to purple. So we know that vestigial to black is 16.5, and then purple to black is 6, right? This doesn't make any sense, right? This number is way smaller than this number. So we know, just logically, we know that that number, or sorry, that arrangement can't be the correct one. So usually we would be able to eliminate one of the possibilities just by the numbers that we have, but then the other two are equally likely or equally possible until we do the actual map and then find out. So what we have to find out is that purple to vestigial distance, because that will give us the answer. Okay. Okay, so here's the same kind of, I won't go through all the genotypes, but I will just point out here are our parental types. They are similar to the two original parents, and then these are our recombinants. And when we take these numbers and we add the two recombinants and divide by the total, we get, I don't know why this is here. That's a mistake for sure. It should be just 0.125 for our R value, or 12.5 map units. Okay, and so the first one must be the correct order, right? Because it's 12 point, and it was not exactly 10.5, but it's closer to 10.5 than it is to 22.5. Okay, so that's what the correct arrangement must be. Purple must be in the middle. Okay, and so that's interesting because this distance now, we calculated for this distance 16.5 map units, right? But if we add these together, this is actually 18.5, right? So something is very unusual here because the numbers aren't really adding up. And so in the next lecture, we're going to talk a little bit more about recombination mapping, and we're going to talk about 3-point crossovers. So that's crosses where you've got trihybrids, genes that are heterozygous for 3 different genes. And we're going to talk about the concept of multiple crossovers. And so before that lecture, and I probably will have some kind of like little exit slip or something in the lecture for this, why do those map distances not add up perfectly? We predicted 10.5 map units for this distance, but we actually got a slightly larger distance. And that means that the outside distance that we calculated between the two furthest apart ones is actually smaller than the number you get when you add the two smaller distances together. One of those numbers is less accurate than the others. And there have been a couple of hints in a couple of places throughout this lecture about what might be going on there. And so just give that some thought. And then when we start talking in lecture 16, we'll get to it. But your learning is always better if you try to figure it out yourself in advance, which is why I'm planting this idea. Okay, we'll see you in class.